it's Dhananjay Garu, Honorable VC sir is joined.
Good morning,
Good morning, sir.
Hi. Oh, you are muted.
Unmute, sir. Hi, Mithila, how are you?
I'm good. How are you? I'm so excited to see you.
Nice to see you. Nice to see you.
Me too. It's been ages.
Very nice.
Maybe decades, probably several decades.
Yeah. Long back. Yeah, very nice.
Long back, IHR days. Yeah, I still remember.
Yeah.
I visited IHR a couple of times. I think you weren't there at that time. You were at IHR.
Yeah.
And then you moved to back here.
Yeah. Sir, so we'll start without wasting much time.
Sir. So, with the permission of Honorable Vice Chancellor, welcome to Dr. YSR Horticulture University 13th Global Connect Series in collaboration with the Conncet State University, Manhattan, USA. The topic is implications of global warming on herbicide efficacy and weed control for sustainable food production. Good morning, sir. Welcome to this Global Connect series. Honorable Vice Chancellor, Dr. T. Janakiram Garu. This is actually, sir, brainchild program from the Horticulture University to arrange the international scientists to give the knowledge on the latest technology of horticulture crops to our teaching scientists, students of all the Horticulture University. All colleges, all race stations, all KVKs, polytechnic colleges are participating in this program. And today's special guest, Dr. Mithila Jugulam, Professor of Weed Physiology, Department of Agronomy, Conncet State University, USA. Welcome, madam. Welcome. Good morning to this program, madam. Respected all university officers, Dean of Horticulture, Dr. Padmavatam Garu, Director of Research, Dr. Naram Naidu Garu, Director of Extension, Dr. Karunasri Garu, Dean of Vijay Studies, Dr. Surya Kumari Garu, Controller of Exams, Dr. Venkaswamy Garu, Registrar, Dr. B. Srinivasulu Garu, Dean of Students, Dr. Salome Sunita Garu, Controller, Madam Shishkala Garu, Estate Officer Mallikarjun Garu, and all associate deans, all zonal heads, all heads of research stations and KVKs, all principals of polytechnic colleges. Welcome to this Global Connect, 30th Global Connect series. Actually, with a lot of encouragement and cooperation from our Honorable Vice Chancellor, we have successfully conducted the 12 Global Connect series, every month one program. And this is the 13th program, sir. Thank you very much. And everyone is getting knowledge of what is happening at the global level. And thank you, sir. Before inviting our Honorable Vice Chancellor, sir, I will give the brief biodata of our speaker, Dr. Mithila Jugulam. Madam is presently working as Professor of Weed Physiology in the Department of Agronomy at Conncet State University. And Madam received BSc Agriculture, MSc Agriculture from APAU, Tirupati campus. Madam is native from Tirupati, and PhD and MSc, PhD and postdoctoral she did at Guelph in Canada. And Madam, after completing MSc, she worked some time at ICR and IHR Bangalore, and along with our Honorable Vice Chancellor sometime. And our present registrar and Gopal Garu, all batchmates, I think. Thank you, Madam, for accepting our invitation. And Madam worked basically in the this weed science, and she is responsible for the teaching of the graduate and undergraduate courses. Presently working as a weed physiologist professor, and especially for the herbicide resistance. And she guided almost 15 MSc and PhD students, 14 postdoctorals, and Madam, her contribution, 90 publications published at a national and international level with the citations of 2000 citations. And Madam has received so many awards, Fulbright Specialist Award, Outstanding Research Award, Distinguished Achievement Research Award, Presidential Award of Merit, and KSU Outstanding Scientist Award, Sigma 11th and Gamma Sigma Delta College of Agriculture Excellence in Graduate Teaching Award, and Jugulam has authored many publications, and she also in editorial board also reviewed so many international journals, and Madam also acted as a member for the several societies. This is the brief report of regarding Madam. Thank you, Madam. Welcome to Dr. YSR Horticulture University. Now I invite our Honorable Vice Chancellor to speak on this occasion. Thank you, sir.
Yeah. Good morning, everyone. I think for Mithila it is good evening.
Yeah, that's okay.
So,
yeah.
Yeah. So, greetings from Dr. YSR Horticultural University. And today we are really very happy that Dr. Mithila Jugulam has agreed to give a talk for our 13th Global Connect series. Just in few words, I would like to mention that this Global Connect series started to connect particularly our scientists and students with global universities and global crop specialists, so that when we are talking about the new education policy, and when we are talking about bringing global universities to our country, I think it is high time that we need to connect ourselves to the subject specialists, and particularly to encourage our scientists and students to take collaborative projects, and also to get connected with the recent research findings at global level. So this is the background of starting this Global Connect series. And I am also happy to mention that two of our guest speakers who participated in this Global Connect series, Dr. Umesh Reddy and Dr. MS Reddy, were recently recognized as adjunct professors to our Dr. YSR Horticulture University. I think just I would like to mention also, we are really happy. I know Dr. Mithila as my colleague when we were working at IHR, and also she is alumni of APAU, erstwhile APAU. I think you studied at Tirupati campus, and most of our colleagues are also known to you. And Dr. YSR Horticulture University is second of its kind in our country. And we have 4 colleges located at Venkatramanugudam, that is main campus, Anantarajpet, and Parvatipuram, and Chinlatrapi. And recently, our university accredited by ICAR, and our university, out of 7 horticulture universities in the country, we got A grade for this university. And we also have 20 research stations working on crop specific and region specific. We have from Rayalaseema, Coastal Andhra, 20th research station, that is at Srikakulam, Peddapeta, we established a new research station, particularly to address the problems of coconut and cashew growing farmers. Am I audible?
Audible, sir.
Yeah, yeah. In between, you broke up a bit, but now you are good. Not now. I can't hear you.
Sorry for that small interruption. So we have even 4 polytechnic colleges, and recently 13 of our varieties got central gazette notification, and also 10 varieties got state gazette notification. And our students are also pursuing their higher studies here and other countries. So definitely this kind of lectures will inspire them for their future endeavors. I think today the topic that Dr. Mithila is going to present is really very important and very interesting, especially for horticultural crops. And when we are talking about the global warming and a lot of climate factors, this is also one of the major researchable area at ICR and also at SAU level. And there is one very popular project, NICRA, is at Krida, Hyderabad. So I think at this juncture, your talk, how this global climatic factors like temperature and carbon dioxide are going to influence this agricultural practices, including weed control, which is a major impediment in agriculture or horticulture. I think we will be definitely benefited. And once again, my greetings from Dr. YSR Horticulture University. Thank you very much.
Thank you. Thank you so much.
Thank you, sir. Thank you very much, sir, for your presidential address on this occasion, 30th Global Connect Series. Thank you very much. Now I welcome our guest speaker, Dr. Mithila Jugulam. Madam, welcome, madam.
Okay.
Proceed.
Sure, yeah. Before I share my screen for my seminar or talk, I would like to thank YSR Horticulture University immensely for this opportunity. When I got the email invite, I was really thrilled and excited. Obviously, I know Dr. Janaki Ram, your Vice Chancellor, well, and then receiving university from my alma mater. Obviously, we were all together when I was a student, and I was really happy to say yes and share part of my research. Again, thank you so much for this invite. I extend my collaboration with your university, and also any students are interested to know more about how the admissions at Kansas State University usually takes place. And if they need any information, they can always contact me anytime via email. So I hope you all can see my slides okay here.
Okay, madam. Visible.
Okay. All good? Okay. All right. Very good. Let me see. All right. So as Vice Chancellor mentioned, I'm going to talk about implications of global warming on herbicide efficacy and weed control for sustainable food production. So as rightly said, global warming is an important issue, not only from US point of view or Indian point of view. Across the globe, people are seeing the effects of the changes in the global environment on crop production. Thereby, we need to look at some of the aspects that we can address for sustainable food production. So here is an outline of my presentation. I'll give you a brief background about my education and research, and then I'll give you a glance about my research at Kansas State University, then focus on global food production needs and what are some of the constraints that we are facing in terms of maintaining sustainable food production. Essentially, my focus will be on weed-related issues. In that regard, I'm going to talk a bit on mechanisms by which we have an important issue, more so in the US, that is herbicide resistance in weeds. That is my area of expertise. I'm going to spend some time on explaining how these weeds evolve resistance to herbicides. And followed by that, I'll talk about temperature stress and how this affects the herbicide efficacy, thereby weed control. And finally, I'll summarize this talk, giving some implications of global warming for weed control. So I come from Andhra Pradesh. I grew up in Tirupati, and I obtained my BSc in agriculture from Tirupati Agriculture College. And then I moved to Tamil Nadu Agriculture University to do my MSc. My MSc was in plant breeding and genetics. You may be surprised how come a person with MSc degree in plant breeding and genetics switched to weed science, entirely a new field. But in my opinion, it is not so. My plant breeding and genetic experience was really helpful for me in getting trained as a scientist at IIHR. After my MSc, I wrote ARS exam and was appointed at Indian Institute of Horticulture Research. That's where I worked with Dr. Janaki Ram for several years, and I still have great memories about my work at IIHR. Great place to work. But what happened was after several years of my tenure at IIHR, my husband got a position in Canada. So I moved, we both moved to Ontario, which is a province in Canada. That's where my husband's job was located. And then since I joined IIHR with only a MSc degree, I wanted to pursue my PhD, and then applied for my PhD program and was selected at University of Guelph. And fortunately, I was grateful to receive NSERC fellowships, both for NSERC is Natural Sciences and Engineering Research Council of Canada, which is an apex agency which funds scientific research, similar to ICR in India or NSF in the US. So they offer several fellowships for a PhD program as well as postdoctoral studies. I received those 2 awards. And how my switch from plant breeding and genetics background to weed science happened was, when I got admission at University of Guelph, my supervisor was very well known in weed science, Dr. Chris Hall, who was my PhD supervisor. He was looking for a person with plant breeding and genetics background to work in weeds. See, he had an idea of looking at genetic basis of herbicide resistance in weed species. So with my background, I was able to get involved in that project. And that was really when I switched my area of expertise to weed science. However, even now, whatever I learned as a plant breeder or geneticist, I'm applying those principles in weed science area. So after working as a postdoctoral fellow at University of Guelph, I got a position at Kansas State University. And then I've been working here since 2011 in the Department of Agronomy as weed physiologist. Currently, I'm also an adjunct faculty at Colorado State University and also University of Nebraska, Lincoln, as well as my alma mater, University of Guelph, and Tamil Nadu Agriculture University as well. In fact, with all these universities, I have several collaborative research going on currently. So before I go into the details of this talk, I would like to thank all my current and past students, postdocs, undergraduates, and visiting scholars who really helped me to establish my lab and make my program a solid one in weed science. I was very fortunate to have folks coming from all over the world. I had in my lab people come from Africa, Brazil, China. Obviously, you can see from this picture, several folks came from India, as well as obviously local US folks were there. And some from Australia also came as visiting scholars in my lab. So working with these diverse groups, I learned a lot from them, their agriculture practices back in their countries. And it was such a great experience to work with different folks coming from different backgrounds. Really, I would like to take this opportunity to thank all of these people, who helped me a lot in my research. And also, I would like to thank my collaborators at the institutional level at Kansas State University. I collaborate with several folks in my department as well as in different departments like plant pathology, Department of Biology. And also, I have established collaborations with several other universities and research, USDA folks, which is like similar to ICR here in the US. You have USDA research centers. I have several collaborations with USDA and different universities. And internationally also, I'm working with folks in Canada, India, obviously with Punjab Agriculture University and TNAU, like I mentioned. And I also have some research going on with Rothamsted research at the UK and Australia as well. And in Canada, I have established collaborations with University of Alberta as well. So these collaborations are really key for my research because as I explain my research, you understand there is multiple disciplines involved. Obviously, with one expertise in your area, you can do multiple things. You have to, in this day and age, it is all about collaboration. Look for areas that you want to expand your research and identify folks with that expertise, and you have to establish collaborations. That was the key, I believe, for my success as well. So if you look at research in my lab, I would say I have three major areas. We do both the basic and applied research. However, more than 80% of my research is in basic work, and I do work with extension folks and I have some applied research as well in my research component. So three major areas of my research is looking at mechanisms, like I mentioned, how weeds evolve resistance to herbicide. So weed resistance to herbicide is a major challenge in the US. If you go and talk to a grower, what's their number one problem? Invariably, more than 90% will say weeds. Herbicide resistant weeds are the major problem here in the US. So my area is, once farmers identify resistance in weeds, we bring those samples to my lab and we evaluate them in the greenhouse. After confirming resistance, what we do is we look at both whole plant physiology and molecular aspects of herbicide resistance. I would say my lab investigates a lot of these mechanisms issues at cellular level. We also do at RNA, DNA, and protein level as well. And that's a major area of my research. And the second area is we are also involved in developing herbicide tolerant crops. In the sense, although you may think it is more like ag industries are focusing quite a bit on developing herbicide resistant crops or tolerant crops, but their focus, if you look at, is only on the major crops like corn or soy or cotton. The industry is focused on essentially the major crops. But in my lab, we are looking at some of the other crops like sorghum. You know, it's a big crop in Andhra Pradesh and also in India. We have looked at herbicide tolerant traits in sorghum, as well as we are looking at wheat. Wheat is an important crop for us in Kansas. Currently, one of my PhD students is working on herbicide tolerant traits in wheat. So that's also another major area of my research. And then the third area, which is the focus of today's talk, is on the environmental effect on herbicide efficacy. So especially we have done quite a bit of work looking at temperature stress and then how temperature stress can influence the herbicide efficacy, thereby weed control. So although, you know, if you look at a lot of our publications come from the mechanism part, but we have done quite a bit of work on looking at temperature stress as well. So for today's talk, I'll focus more on this, although, you know, if anybody is interested to learn in other two areas, feel free to contact me. I'm more than happy to share the research in those areas as well. So as you all know, it is being projected that global population by 2050 is expected to increase by 2 billion people. So therefore, our requirement as a scientist is, or I would say our responsibility as a scientist is to meet the needs for increasing population in terms of food production. So there is a need of 60% increase in food production considering the increase in the global population. However, we need to be aware of some of the crop production constraints. I have listed only 4 here, but obviously there will be many more. But these are some of the important crop production constraints, like input costs, as you agree. The input costs are constantly getting a burden on the growers. They are increasing every year. The costs are increasing. That's a major challenge. And also water availability. It's not a one region specific problem. It's a global issue. Agriculture obviously depend on water, and water resources are depleting. That's another major constraint. And then importantly, climate. So with the change in climate over the last several decades, we see the crop production being influenced by change in the climate. It's like Dr. Jankiram mentioned, the fluctuations in carbon dioxide and temperature as a result of global warming is going to be a great challenge for future crop production. And not last but not the least one is the pest. Pest infestation can also influence the crop production significantly. So if you look at climate, like I said, I'm going to focus on temperature changes. As you see here, global projections in terms of temperature increases have been already noticed over the last several decades. So with this change with this increase in temperature, generally crops or plant growth and development has been affected negatively. I put a question mark there because weeds are also plants. Sometimes the changes in temperature or carbon dioxide, we have seen it's going to really benefit some of the weeds that are like C4 weeds. So obviously, we don't want weeds becoming more aggressive with the change in climatic changes. So how can we address that issue? If crops are not doing well and on the other hand, weeds are doing very well, we don't want such scenario, right? We have to address those issues. And also it's been projected that quality and quantity of crop production will be affected with the change in climate, or especially with the temperature increases. So one aspect I'm going to talk about is temperature stress. Obviously, it's going to be on the pest, which is my focus of the pest is weeds. So we have several agriculture pests like insect pests, pathogens, which are like diseases, and more importantly, the weeds are going to be the major pests of agriculture production, and crop losses due to weeds have been projected to be greatest among the other pests. Like if you do not take proper control measures, growers can end up having more than 45% yield loss because of weed competition. However, in this regard, chemical weed control has been found very successful. As some of you may be aware, during Second World War, the first chemical that was synthesized was 2,4-D, which was eventually used as a herbicide for broadleaf control in agriculture systems.
Hello?
In cereal crops, successfully it was used, and we have seen this revolutionizing the agriculture production. So I just want to mention here, I know you are all in horticulture, working in horticulture crops, and your focus is in horticulture crops. But I think everybody here, whoever has joined this seminar, agree weeds are also an important component or important pest in horticulture production systems as well. So they are everywhere, and we need to address this issue. So once the chemical weed control took up the kind of scenario of weed management in many countries, especially in countries like US, what happened was almost the industry also focused on developing herbicide resistant crops. So in US, if you look at majority of major crops like soybean here shown, or corn, or corn, or cotton and canola, they all have herbicide resistant traits. So once farmers adapted to this herbicide resistant crops, fields started to look more like pristine. There is no weed, and it was almost like a silver bullet for growers. They grow these crops, they apply the herbicide, and they manage the weeds. So it was literally a silver bullet for growers once they adapted to herbicide resistant crops. However, it didn't last long, right? Weeds strike back. Obviously, the selection, because of repeated use of the same herbicide, has resulted in evolution of resistance to herbicide. So if you travel across the Midwestern US, Kansas is part of Midwest, fields looking like this are not uncommon, infested with herbicide resistant weed. Sorry about that. That's why I said herbicide resistance is a major issue in many farmers' fields here in the US. So this challenge has been continuing, and growers are looking for some options to manage these weeds. So if you look at these graphs I show here, the top one is global increase in herbicide resistant cases. So as you can see, after 1980s, there is a steep increase in the number of cases of herbicide-resistant weeds globally. And the situation in the US, shown in the graph below, is not any different from global scenario. Here also, we have seen evolution of resistance in weeds to herbicides has increased tremendously. And then if you look at, you may come across several definitions for herbicide resistance. I'm using one here, very simple one, which is loss of effectiveness of a herbicide on weed population. Basically, if you do not see control of weeds by a particular herbicide, once it used to be a good option for controlling that weed, and then you see the resistance evolution must have occurred in that population. So herbicide resistant, I want especially students in this seminar understand that it's a naturally occurring phenomenon. It occurs due to selection. Herbicides do not cause any changes in the plant, or they do not induce any mutations for the plant or weeds to evolve resistant. It's a naturally occurring one. It's because of natural selection. So if you apply the same herbicide over and over again, you end up selecting for the individuals that are resistant. So here in this cartoon, I'm showing, so in a population of weeds, like I imagine this is 100 weed plants, there will always be one plant that has naturally some characteristics to withstand herbicide application. So if we repeatedly use the same herbicide over and over again, obviously this has the resistance trait. So this particular plant will survive the herbicide application, grows and produces the seed, reproduces and produces seed. See, next year, the one shown in black is the resistant plant. Next season, you see increase in the number of herbicide resistant individuals. And again, subsequently, if the farmer uses the same herbicide again, and the resistant plants will dominate in the field, thereby you see complete takeover of weeds in the crop production system. This is how the evolution of herbicide resistance occurs in nature. So basically, we need to identify very beginning itself if there is resistance, then take control of these populations before they become a big problem in the field. So having said that, how do these weeds evolve resistant? Like what are the mechanisms by which these weeds evolve resistance to herbicide? So in weed science, we group them into two major categories. One is the target site resistance. As the name implies, I guess you all know, each herbicide has a specific target in the plant. A target is essentially, in most cases, an enzyme or a protein. So basically, if any change or any mutation in the gene coding for that target protein occurs, then what happens is there will be a change in the amino acid sequence, and thereby the protein structurally, it changes the folding of the protein, thereby the herbicide cannot bind to that protein anymore. So the mutations in the target gene results in the protein alteration, therefore herbicide cannot bind and the plant will be resistant to herbicide. Again, another target site resistant mechanism is it could be because of the gene amplification, which means the target gene is duplicated several fold. We have seen in some weeds like palm amaranth, which is a major weed in Kansas or throughout the US, I would say. Resistance to glyphosate or Roundup is as a result of amplification of the gene. So which means the resistant plant had about 100 times more copies of the target gene. Therefore, you have more enzyme available and the plant can withstand herbicide application and survive. So that's another mechanism by which the weeds can evolve resistance. Or sometimes without amplification also, you can have increased gene expression because of some promoter alterations. So we have seen some cases where you have increased gene expression resulting in herbicide resistance as well. So all these mechanisms are grouped as target site resistance. And the other type is non-target site resistance. So as the name implies here, the herbicide target is not affected. A herbicide target is still sensitive to herbicide. However, there are certain mechanisms by which herbicide gets inactivated before it reaches the target. So it could be, it could be because of reduced herbicide absorption or uptake, or translocation. Translocation is movement of herbicide within the plant. So for a better weed control, you need to have herbicide being translocated throughout the plant. Therefore, every cell in the plant will receive the herbicide, and you see the herbicidal activity. So you may have reduced herbicide absorption or translocation, resulting in herbicide resistance, or in many cases, it is because of herbicide metabolism. What is metabolism means? Herbicide gets deactivated or degraded. So there are certain enzymes in plant, such as probably you heard about cytochrome P450s. So they are the enzymes. Even human beings, we also have several cytochrome P450s. The role of these enzymes is just to fight against foreign molecules. So they are evolved in evolutionary trajectory. All organisms evolved the cytochrome P450 enzymes to fight against any foreign molecule entering the system. So here in the weeds that are resistant to herbicides via metabolism, what happens is the cytochrome P450s is involved in degrading the herbicide molecule, making them inactive. So what I'm showing here is the peak shown here in the red is the active form of herbicide. Once it is metabolized, you see different peaks. That means the herbicide is metabolized. These chromatographs we obtained by using radio-labeled herbicide running through a high-performance liquid chromatography or HPLC analysis. So these are the two major mechanisms by which weeds can evolve resistance. So if you have target site resistance as a major mechanism, then in a way it's good that it does not confer resistance to other herbicide targets, which means if you have a target site resistance in a weed, you can change the herbicide mode of action, or you select another herbicide, then the weed may be still susceptible to the new herbicide. So that is a method that we can use for weed control. Whereas if you have a metabolism-based resistance, what happens is the plants are predisposed to evolve resistance to other herbicide targets as well. In fact, we are seeing more and more cases of metabolism-based resistant herbicides in our major weeds. That means growers will be losing opportunities or options to manage such weeds because they've already predisposed to evolve resistant to other herbicide targets as well. So if you ask me, if a weed has a metabolism-based resistance, that will be more challenging for weed control rather than the target site alteration. At least here we have other options to manage the weeds. Okay. So now switching the gears, I'm going to talk about on the temperature effect on herbicide efficacy and also herbicide resistance in weeds. I'm going to give you several examples and the work that we have done in my lab. I'm listing here 4 weeds here. One is the Palmer amaranth, which is, a major weed in not only in the US, it's a major weed across the US, not only in Kansas, across the US. This is the Latin name is Amaranthus palmeri. I know in India, Amaranthus species are grown for grain purpose. They are like crops. There we I think a lot of area is increasing for grain amaranth production. However, this Palmer amaranth is a big weed in US system, and a single Palmer amaranth plant can produce up to 600 to 700 seeds under ideal conditions. So that's another major challenge. Once you have a resistant plant in the field, if it survives and produces, it can have a lot of seeds, resistant seeds in the seed bank. In the next season, you will end up having a huge problem. Another weed is called kochia. its Latin name is Bassia scroparia. So this weed was introduced in US as an ornament, but eventually it invaded, and it's a major problem in Great Plains, US Great Plains. Basically, western part of the US, this weed is a huge problem, including western Kansas. In Kansas, this weed has become a major challenge for wheat growers, even cotton, corn, and sorghum growers as well. Another weed is the common water hemp, or in short, water hemp. This is also an amaranthus weed, Amaranthus tuberculatus. Both Amaranthus palmeri and Amaranthus tuberculatus, they both look pretty similar. If you closely look at them based on the leaf shape, you can distinguish. However, water hemp is a huge concern in the eastern part of the US, whereas Palmer amaranth is a big challenge throughout the US. And we have another weed called common lamb squatters. I'm sure this is a weed in India as well. It's a Chenopodium album. We call these as driver weeds. Driver weeds, you may ask, why are they called driver weeds? Because these days, these weeds are deciding the crop production decisions. In the sense, farmers are so concerned about the presence of these weeds, they are changing their crop production decisions based on what type of weeds they have in the field. They are driving the crop production decisions. So really, it's very unfortunate situation, but that is the severity of the weed problem that we are facing in the US. So what we did was we looked at Palmer amaranth, shown here, and temperature effect on Palmer amaranth control using mesotrione. Mesotrione is a herbicide belonging to HPPD inhibitors. HPPD is an important enzyme in carotenoid biosynthetic pathway. Basically, if you use the herbicides like mesotrione, it's a trade name is Callisto. I'm not sure if it is registered for use in horticulture crops. I know in India it's been registered for use in corn, I believe. So Callisto, how it kills is once it's applied on the weeds, the sensitive plant, the herbicide reaches the chloroplast and it binds to the enzyme HPPD, and thereby the downstream reactions are inhibited, resulting in carotenoid depletion. Therefore, the plants will turn chlorotic and eventually necrotic and die. That's how the herbicide mesotrione works. And what we did was we grew Palmer amaranth plants at three different temperatures: low temperature, optimum temperature, and high temperature. As shown here, low was 25 and 15 day and night, and optimum 32.5 and 22.5, and high was 40 and 30 Celsius. So as you can see from the pictures, like they say, pictures tell 1,000 words. As you see here, when we grew Palmer amaranth plant at low temperature, if you look at the plant treated with approximately 26 grams of Callisto or mesotrione, the plant is completely dead. Whereas if you look at the plant grown under high temperature, even we applied double the dose of 26, which is about 52 grams, the plant is still surviving, although there is reduction in growth compared to non-treated or low herbicide dose treated one. But the bottom line here is, if you grow plants under low temperature, you just need little herbicide compared to those grown under high temperatures to kill the plant. In fact, the field recommended dose of mesotrione is 110 grams. So you see, we are using only 26 grams, but still you can kill the weed. The take-home message from this photo is that if the plants are grown under low temperature regimes, you can see a better control of this weed than when they are grown under high temperature. This was also documented with various parameters that we recorded in plant height, chlorophyll index, or visual injury, as well as Fe by Fm is the photosynthetic efficiency. All these parameters indicated that at low temperature, weeds after treatment of herbicide or better control compared to those grown under high temperature. So then we asked a question. So what's going on when plants are grown under low or high temperature when you apply this herbicide? What exactly is making these amaranthus plant to withstand herbicide application at high temperature? So as I explained in my mechanisms slide, what we did was we looked at herbicide metabolism, a non-target site, like, it is a kind of, it's a more common one. Mesotrione is known to be metabolized in corn where it is registered for use. So we tested the metabolism of mesotrione in plants grown under low temperature as well as high temperature. And as you can see here, on low temperature regimes, the peak here shown with the mesotrione structure. I'm sorry, the mouse is not doing that. I'm not doing. So you see here, this is the parent compound or active compound of mesotrione. You see, if the more mesotrione is present in plants grown under low temperature, that means these plants are receiving active form of herbicide, and thereby they are not performing well when they are grown under low temperature. Whereas at high temperature, you see the active form of mesotrione is very less. That means it is degraded or metabolized into inactive form when the plants are grown under high temperature. Again, we show this in this graph as well, the amount of parent compound. Parent compound we mean is the active form of mesotrione or the herbicide that can kill the weed. So again, at low temperature, you have high amount of the herbicide available for doing the herbicidal activity, where compared to the one at high temperatures, you have much less of the active form. Basically, what we found was enhanced metabolism of this herbicide mesotrione at a high temperature controls for poor control of palmer amaranth when they are grown under high temperature. So we were also interested to see the target site mechanism too, like is there anything happening with the HPPD gene, which is the target of this herbicide. So we looked at the gene expression in plants grown under different temperatures, again listed here, same as the previous slide. So when we looked at the expression of the palmer amaranth HPPD gene grown under high temperature, as you can see from this graph, at high temperature, after 48 hours of the treatment, the HPPD gene expression is increased several fold. So that means the plant is making more of this enzyme. So even if you have the herbicide applied, the enzyme is not affected. So basically what is going on in this story is that the poor control of mesotrione, palmer amaranth with mesotrione at low temperature is basically, you have two things going on. Herbicide is not getting metabolized and there is not much herbicide, target gene expression, which is the HPPD gene expression. In other words, at high temperature, you have rapid metabolism of the herbicide as well as increased gene expression. These two are contributing to the poor control of the weed using this herbicide. So then what we have done is we also have some populations of palmer amaranth that are found resistant to this herbicide. So previous slides I showed you a sensitive palmer amaranth plant, which is known to be killed by this herbicide mesotrione. So then what we did was we looked at, is there any difference in the resistance level when plants, susceptible or resistant plants are grown at high temperature or optimum or low temperature? Again, there is same story here. So at high temperature, you can see both susceptible and resistant plants when treated with herbicide, they are not affected at all, as opposed to those grown under low temperature. So you can see susceptible plants showing the bleaching symptoms. Like I said, that's how the mode of action of this herbicide, you see the bleaching symptoms. And both resistant and susceptible grown under low temperature are becoming very sensitive to this herbicide. So basically, the resistant plants, if you grow them under high temperature, they are doing perfectly fine, even susceptible ones. That's the same scenario. You are increasing the resistance if the plants are grown under high temperature, and if you want to see better control of Palmer amaranth, both mesotrione susceptible and resistant, you need to use or apply the herbicide when the temperatures are cooler. Basically, what the message, take-home message here is, we are suggesting the growers for this herbicide, if you want to see better control of this weed, Palmer amaranth, you better apply when the weather is very early in the morning, when the temperatures are cooler, then you see better control. In fact, our results were translated to the growers in a very simple manner through our extension specialists. They showed our work to growers, and growers reported that they applied this herbicide at cooler conditions. They have seen better results. So it's been actually seen under field conditions as well. So then I'm going to switch a different herbicide again in the same weed, Palmer amaranth. This work was done by Eduardo Riddell. He's from Brazil. In fact, he was an intern in my lab for 6 months. He came as an undergraduate intern, but has done excellent job in such a short time. In fact, we are he got a nice publication also coming out of this work. I was very pleased to have him in my lab as an undergraduate intern. So what Eduardo did was he, before I go into the details, I just want to give some background about this population of Palmer amaranth that we used. So before Eduardo used this plants for his work, my other student, Chandrima Sham, looked at 2,4-D resistance in Palmer amaranth. So the first case of 2,4-D resistance in Palmer amaranth was reported from Kansas, and we have actually characterized the mechanism of resistance. That was Chandrima's work, where she looked at why the resistance in 2,4-D in Palmer amaranth was evolved. She confirmed that 2,4-D resistance is because of metabolism of 2,4-D. So we already had that first-hand information. Then Eduardo wanted to look at what will happen to the 2,4-D resistance under temperature stress. So what he did was he grew 2,4-D susceptible and resistant plants under 2 different temperatures: low temperature 24 and 14 day and night temperature, and high temperature 34 and 24 day and night. So if you look at the susceptible plants on the top panel here, grown under low temperature, and resistant plant grown under low versus the bottom one, high temperature. So when the plants are grown at low temperature and treated with various doses of 2,4-D, you can see the resistant and susceptible plants are much more affected when they were grown at low temperature. Whereas high temperature plant, look at the resistant plant, they are doing really well even after treating with 2x. 1x means is the field recommended dose, which is 560 grams for 2,4-D for controlling this weed. So we went up to 4 times field recommended dose. Still the plant is not completely dead, grown under high temperature, especially the resistant plant. So basically, the plant biomass has increased when they were grown, regardless of susceptible or resistant. When they are grown under high temperature, the biomass of Palmer amaranth plant is increased, whereas those that are grown under low temperature, you see decreased biomass. So similar to the previous story where we show mesotrione also Palmer amaranth also performed very well at high temperature when treated with mesotrione. Here also the same story. Again, we looked at the mechanism why plants are doing better when grown under high temperature after treatment with 2,4-D. Again, these are the chromatographs showing the herbicide metabolism, like 2,4-D metabolism. As you can see, plants grown under low temperature. Here, the MSS means this is the susceptible plant. KCTR is an acronym for the resistant plant. So in the susceptible plants, you see a lot more herbicide 2,4-D being active at low temperatures compared to high temperature. As such, the resistant plant is metabolizing 2,4-D. Like I said, my other student work confirmed the metabolism of 2,4-D in resistant plant. But what happened was at high temperatures, there is actually not much change at all. It's still active, and you see the herbicide getting more active on the Palmer amaranth plant. So basically, again, the take-home message here is at low temperatures, even Palmer amaranth, if we have 2,4-D resistant, they can be controlled, both susceptible and resistant plants better when they are grown under low temperature regimes. So then I'm going to give some information about kochia weed. Like I mentioned, this was introduced as an ornamental plant from Europe, and very soon it became a weed. My second PhD student, Junjun Oh, currently is working as a herbicide discovery scientist at Corteva in Indianapolis. Very, very good student, great student. He has got 6 publications from his PhD. Very productive student, very pleased to have him in the lab. So Junjun looked at kochia, this weed, and he worked with glyphosate and dicamba. These are the 2 herbicides. I'm sure they both are registered for use in India also. Glyphosate is a very widely used herbicide, and it inhibits an enzyme called enol pyruvate shikimase phosphate synthase, EPSPS, in shikimic acid pathway, whereas dicamba is an auxinic herbicide or synthetic auxin herbicide similar to 2,4-D. They are known to work as a plant growth regulators. High doses of dicamba 2,4-D will result in abnormal plant growth and eventual death. So here what Junjun did was he looked at temperature effect on dicamba efficacy. So the kochia, if it is a sensitive plant, it's pretty easy to control with dicamba. In other words, dicamba is very effective on kochia to control them. So we grew the plants at low and high temperature, the dicamba plant, and treated with various doses kochia plants, and treated with various doses of dicamba. As you can see, when plants were grown under low temperature, even 0.5x, which is half recommended dose of dicamba, can kill these plants very easily. Like even the 1/4 of x plants show very, the growth is still good, but at 0.5x beyond that, you see better control of this. Whereas when we grew them under high temperature, we have seen even 1x plants surviving well, and there was increase in biomass. Basically, the plants grown under high temperatures, you see increased biomass, whereas low temperature plants can be controlled well. So again, our goal is what's happening. Why are this type of response we are seeing in kochia grown under different temperatures? So when we used the radio-labeled dicamba and looked at dicamba absorption and translocation, you can see here the absorption of the kochia plants in terms of dicamba uptake is pretty similar at all 3 temperatures that we tested. Whereas when we looked at the translocation pattern, we have seen much less herbicide getting translocated in plants grown under high temperature. Obviously, this is a clear indication that not enough herbicide is moving throughout the plant when plants were grown under high temperature. It's a different story than what I talked about in the two other cases, which was metabolism based, but here it is decreased translocation. Basically, in plants grown under low temperature, there was increased translocation, whereas plants grown under high temperature, there was reduced translocation. So if the herbicide is not moving throughout the plants, obviously it's not going to be effective in controlling the weed. And then Junjun also looked at effect of glyphosate, basically under two different temperature in controlling kochia. Again, we have seen reduced biomass in plants grown under low temperature and increased biomass in plants grown under high temperature. In other words, plants did well when grown under high temperature. But here, that story is different than dicamba. So as you can see in this graph, where we show the amount of herbicide absorbed, which is basically, you can see the difference in kochia plants absorbing glyphosate at 3 different temperatures. So basically, in plants grown under low temperature, there was less glyphosate absorbed. Therefore, there is not enough herbicide reaching inside the plant to have the effect of glyphosate. So it's a 2 different mechanism. For dicamba, it is reduced translocation, whereas here it is reduced absorption. In other words, you have at low temperature better herbicide uptake, therefore the plants can be controlled very well. So again, for each herbicide, although the mechanism is different, but ultimately the response is the same, which is at low temperature, you see better weed control. It could be, the mechanism could be anything, but overall the effect is the same. Again, we have looked at another problem weed, which is common water hemp. Here, Chandrima, like I mentioned, she graduated last year with PhD. Currently, she is working for Bayer Crop Science in their herbicide physiology division. So she looked at water hemp, which is also found resistant to 2,4-D. Actually, this population came from our neighboring state, Nebraska. Like I said, I collaborate with folks in Nebraska. They asked us to investigate what is happening in the 2,4-D resistance in water hemp. So Chandrima looked at the effect of temperature on 2,4-D resistance in this weed as well. Again, similar to what I showed earlier in Palmer amaranth, she grew the plants at low and high temperature. Again, at low temperature, you see better control of both susceptible or resistant plants, and high temperature plants are doing pretty good, even at 6 times or 8 times the field recommended dose, especially the resistant plant. So biomass proportionately increased even with the several doses of herbicide that we use. And then she also looked at the mechanism. Here it is similar to what I explained to you in Palmer amaranth. It's metabolism. At high temperatures, the plants are able to rapidly metabolize 2,4-D. Therefore, not enough herbicide is available for having the herbicidal activity. So basically, the amount of herbicide being active is reduced at high temperatures compared to low temperature. So then we also looked at glyphosate effect on common lamb squatters, Chenopodium album. And this was done by my master's student, Randy DeGreef, and my then postdoc, Aruna Varanasi. She's also with Bayer now, and Randy is working as an agronomist in Minnesota. So they have looked at effect of glyphosate on lamb squatters grown under different temperatures. So again, same story, low temperature, the plants are controlled very well compared to those grown under high temperature. So basically here, lamb squatters plants, if you grow them at low temperatures, you can control them with lower amount of glyphosate than those grown under high temperature. The field recommended dose of glyphosate is 840 grams. I'm not even talking about resistance here. These lamb squatters are just susceptible plants. So when they are grown under high temperature, even double the field recommended dose did not kill the plant. So this is really a challenge. There are temperature fluctuations occurring throughout the crop growing season, especially in places like Kansas. We see a lot of fluctuations during crop growth period. And depending on the herbicide they are using or the weed species they are targeting, farmers should be really careful in getting better control. So basically, the summary from this part of my research is, with the increasing temperature, what we anticipate is weeds can become more aggressive, and basically there could be some weed shifts occurring, because once you see the shifts of weed, then farmers need to really focus on changing their weed control tactics. Their programs, depending on the weeds and the crop they grow, will all need to be altered accordingly. And also weed seed bank dynamics will also change. Therefore, in the next season, you will have much more challenging weed dynamics for management, especially if you are looking at resistance, herbicide resistance. It gets even more challenging with increased temperature regime. So having said that, this is really good information we generated, but I believe there is a lot more research needed, even to look at other herbicide mode of action, other weeds, and also understand what is going to happen in terms of weed ecology and management that we need to focus on to recommend good management practices for growers for future climate condition. But I believe in this research, we are still making baby steps, but there is a lot more to do. NIFA, our funding agency, they are also now calling for proposals focusing on climate change, and we are fortunate that this call came recently, and we are interested to submit a proposal with our preliminary work that we did. Hopefully we can take the research to next level. With that, I would like to acknowledge all my funding agencies. As you all agree, without the support from your funding agencies, we can't do much work. I'm fortunate to receive funding from several agencies, and we could do some good work in this area. With that, I'll stop and take any questions you may have. Thank you again for your patience.
Thank you, madam. Excellent, excellent presentations. And in global warming scenario, how the temperatures are affecting weeds, and how the mechanisms of weed resistance to herbicides, development of herbicide tolerant crops, environmental stress, and herbicide efficacy. And you have also covered 45% of the weeds affecting the yields in crops, and 30% insects, the 20% diseases, and evolution of herbicide resistant weeds. And you have covered many mutations in herbicides, target gene amplifications, and how that reduced absorption of translocation and herbicide metabolism. And so you have covered so many crops also, like Palmer amaranthus, Kochia, common water hemp, common lamb squatter. These are the weeds, how they are affected, and how they are tolerant to herbicides, and what are the metabolites like mesotrione and PD gene expression, and dicompa. These are the metabolites, how they are affected at low temperature and high temperatures, how the tolerance is developed. Excellent presentation, madam. Thank you very much. A wonderful talk on this weed physiology. Now I invite our Honorable Vice Chancellor to interact.
I think we should take some questions.
Yeah.
Yes, sir.
That's important.
I invite questions from the audience, please.
Madam, good evening, madam.
Yeah, good evening.
Yeah. Madam, in this context of development of herbicide tolerance, what are the alternate approaches you can see for the weed management?
You
Whether
mean,
it is a weed side or whether a fungicide, over a period of time they are losing their efficacy, and the crop protection or like weed management is becoming very highly challenging.
correct?
So what
What's
are
the
the
question?
what are the
You
other
are asking-
what are the other alternate approaches what you can suggest apart from rather than depending on this?
Okay. Yeah, you are asking how can we manage weed without only dependent on herbicides? What other methods we can use? Is that
Yes,
correct?
yes, ma'am.
Okay. Yeah, that's a very good question. See, these days our mantra is also don't only depend on chemical weed control as a single strategy to manage your weeds. What we are promoting is more like an integrated weed management. We are recommending growing cover crops to reduce the weed pressures to start with, and also we are also suggesting obviously the crop rotations and include diversified crops. And we are also promoting kind of, although it's not very well researched yet, but biological control is also another option we are looking at. For now, we want mechanical, some level of mechanical weed control also. So if you look at US, we mostly depend on no-till agriculture, right? That's a big part of our crop production system. We do not, especially to conserve moisture and soil. This has been going on for several decades in the US. No-till agriculture is really, really critical. But if a grower is going for no-till agriculture, invariably he needs to depend on herbicides for weed control, because in no-till agriculture, you do not have option of tilling and getting the weed seeds exposed to the top layer so that they don't germinate. That's not there. Thereby you see weed pressure increasing in no-till agriculture. So they depend on the herbicide. So what we are suggesting in the integrated weed management practices is some level of shallow tillage, at least, especially the weed that I talked about, kochia. They are pretty shallow in the sense they are located on the top 1 or 2 centimeters. So if you resort to shallow tillage, you can at least manage many weed species that are small seeded. So these are the holistic approach we are suggesting rather than depending on only herbicides.
Okay, thank you, ma'am. Actually, you were telling about that biological control. Actually, that was where I was personally feeling that will be a better approach, because initially, like when I was working at banana research station, there was some organic farming plots, and the majority of the investment was going for the weed control. And there was, because it is organic farming, there was no application of any glyphosate like that, and majority of the labor was going to for that weeding only, manual weeding. So at that time, I saw some of this is a cypron or this one. I saw some of the rust infecting these weed species like cypress, I think, or cyanodon. So I thought like whether if we can, because if they are host specific, can we multiply those in a glass house, and if we spray that will be a better approach, I felt. So, and you were mentioning about biological control. If you can see those, if they are not if they are not affecting crops also, that will be a better approach of using biological control if they are specific to particular those weeds. Then there will not be any development of resistance or like this type of lot of hindrances.
See, that's really true. But the issue, like you mentioned, specificity. whole specificity of these biological agents is really a challenge, right? You don't know how they are going to affect the other non-targeted ones. So that has been a challenge in developing biological based weed control. In our university, in the Department of Entomology, some work was done, but it hasn't reached to the farmers level. It's all in the lab still because of the lack of complete specificity in terms of controlling. But there needs to be a lot of work that needs to go in this area. Currently, we don't have enough data to say one particular insect is good on palmer amaranth only, or in dicot weeds versus monocot weeds. So that's the challenge. But that's something we need to invest and look in detail and see what we can come out with. If we get a very specific insect that can control dicot, then that's great in cereal crops, right? Those are the things we are still not at that stage to make it to applied, or in application in the field. I see a chat. I think Madhvi says rotation of herbicides is an option. Of course, yes, that is absolutely true. When I said crop rotation, which also involves herbicide rotations also, because we have very specific herbicides registered for each crop. So obviously, if you do crop rotations, you also rotate the herbicides as well. So that's an important strategy. Thank you, Madhvi.
Thank you, madam.
Madam, you are
Anybody?
telling that you are collaborating with other departments like pathology. Like, can you throw some light on what exactly it is on how you are collaborating with it? How you are using it in your area of expertise?
Okay, that's a very good question. So, like I said, I have focused my talk only on my third area of research, right? Environmental stress and herbicide efficacy. My major area is on mechanism of resistance. So I collaborated with folks in plant pathology, especially Dr. Bikram Gill. I'm sure some of you may heard about his name. He was a wheat geneticist. He is a pioneer in molecular cytogenetics area in the world. They are the ones, their group developed the technique like fish fluorescent in situ hybridization, GISH, genomic in situ hybridization techniques. I collaborated with their group to look at molecular cytogenetics aspects of weed resistance in particular. Using that technique only, we learned I mentioned about gene amplification, right? In palmer amaranth, glyphosate resistance is because of amplification of the target gene, like 100 copies we have seen. All that work and using molecular cytogenetics, we actually showed where exactly on the chromosomes these copies are present. So that is a very unique expertise and unique technique that we can use to physically show the genes on the chromosome. In that regard, I established with plant pathology a very solid collaboration, and we were able to publish in PNAS, Proceedings of National Academy of Sciences, several papers, plant physiology. All that great work came because of that collaboration. So that's what we did with the plant pathology folks.
Thank you, ma'am. It was very pleasant listening to you taking different crops and different fungicides, these model systems.
No, thank you.
Thank you.
Thank you, Karen. Next, anybody?
There is somebody raised their hand. Shrikant Dasari.
Hello? You can speak.
Oh, sorry. I don't think that's...
From University of Research Journal Heads, Scientists.
Are there some
If
students
nobody-
also?
Students also there.
Okay.
Anybody, you can ask questions, madam. If nobody is there, I invite our Honorable Vice Chancellor.
So it's really great. Congratulations for that outstanding presentation,
Thank
Dr.
you.
Mithila. And also your great team of students. Really
Yeah.
a wonderful work. Yeah. I think you have really clearly given message that how this climate will play an important role in weed-side efficacy. And the take-home message is that the herbicide efficacy and it can improve at lower temperatures. And of course, interestingly, you have mentioned that 45% losses occur due to weed.
Yes.
Rather than pests and also diseases. Around 20% from the diseases and insects, you mentioned 30%. So when we are talking about the sustainable agriculture, I think I can take example of onion. More than 60% of the labor is utilized for weed control manually in case of onion.
Wow.
So I think you also clearly sent a message that being a plant breeder and working on agronomic aspects, and here also I remember have come across the research physiological basis for breeding. So where much of the work carried out in coffee, and that has tempted me to study in marigold, and I have published 2 papers in that. So this is really one more clear message that anywhere science will contribute.
Yes.
And particularly, you have given how the temperature regimes are increasing from 1951 to 78. I think it is very alarming at this situation that this kind of studies will contribute much more for further, research and also practical implications in this area. And lastly, I really appreciate that you have told the results are taken to the field level and through your extension workers. And really, this is the need of the hour. When you mentioned that even a small suggestion that whenever they take up this herbicides application, it should be in the morning. That is where the small interventions and suggestive measures will really help the farming community. I think this is to be taken as a note. Of course, really we can see in future that we can explore for collaboration in these area between Kansas and Dr. YSR HU. And we can, and lastly, I have one worry. When we are applying these herbicides or weedicides to the soil, so whether anything has been worked out, how these useful microbes also getting affected, whether whether all these also contributing in the scenario of climate change and whatever your research area?
Yeah, so that's a good question. And currently this semester I'm teaching a class on herbicide interactions. To answer your question, in my lab, we have looked at soil applied herbicide, not in terms of microbial activity, but just look at soil applied herbicide versus post applied herbicide in terms of resistant mechanism. So that's a different story. But to answer your question in terms of microbial interaction, in fact, in wheat science, there is one phenomenon called enhanced degradation. Okay. So what happens here is, if you apply, especially the herbicides like 2,4-D or atrazine, they are prone to expose to this enhanced degradation. What it means is they are applied to soil. So the microbes will also work on herbicide for their nutrition. So herbicides are organic molecules, right? They have carbon, hydrogen, and oxygen. So the microbes use those herbicide molecules for growth and development. So they continue to grow and multiply. As you know, microbes their life cycle is 20 minutes, right? Literally, like they multiply many fold. So next season, farmer applies the 2,4-D or atrazine, but we have in the same soil millions of those microbes. Right away, they go and act on that herbicide because they are used to degrading this herbicide. That means there is not enough herbicide for the purpose grower applies, which is weed control. All the herbicide is taken up by the microbe, so which we call as enhanced degradation. That is a very well established phenomenon. But what happens to microbial degradation in terms of changing climate condition, I don't think anybody has published, to my knowledge. Maybe some folks are working, but I am not aware. But that's a very interesting area to look at. But as such, enhanced degradation is a huge problem, especially with atrazine, which is very widely used in the US as a soil applied herbicide. So we have seen many cases of enhanced degradation. But your point on climate change and microbial degradation, I haven't seen any publication. Probably there is going to be significant effect in that area also.
So
Thank you, sir.
really thank you, Mithila.
No problem. You're welcome. I really enjoyed. Again, I'm like I said, I'm happy to collaborate in any capacity with your university. Let me know how I can help. Maybe we can exchange like students or bring some student and they can get trained in my lab, or I can connect you guys with some horticulture faculty. And we have a very big horticulture department.
Sure.
Opportunities are plenty. We can start some collaborations. Yes.
Yeah, sure. That's very nice, and thank you.
Thank
And,
you. Thank you all.
Nice to hear you.
Yeah.
Thank you, sir.
Yeah.
Thank you, sir, for your remarks, valuable remarks. And thank you very much. Now I invite our Registrar Garu, Dr. B. Srinivasulu Garu, to say a formal vote of thanks.
Thank you, Dr. Dhananjaya, Director Industrial and International Programs, Dr. YSR Horticulture University, for giving me the opportunity to propose formal vote of thanks for this wonderful program. Today we have, on behalf of Honorable Vice Chancellor of Dr. YSR Horticulture University, Dr. T. Janakaram Garu, and my colleague, Dr. Dhananjaya Rao Garu, and my colleague university officers, and all the participants who have participated in the 13th Global Connect Series program. And on my own behalf, I extend my heartfelt thanks to Dr. Mithila, who is also happened to be my colleague. Today, I feel very proud to hear you on this wonderful talk, what you have given on the subject implications of global warming on herbicide efficacy and weed control for sustainable food production. Really, it is very excellent and nice talk on this topic, which has really enriched the knowledge on this, particularly the environmental stress and herbicide efficiency on which you have worked. My compliments to you for this commendable work you have done, particularly on these weeds and the global, in the context of global warming. I'm extremely thankful to you for your excellent presentation with a lot of illustrations you have given with examples and very, very clear. The presentation is also very clear with good photographs. I really, extend my heartfelt thanks to you for your wonderful talk on today's program. Thank you, Dr. Mithila, for being with us almost 2 hours with us, spending your valuable time and sharing your, experiences with all our faculty members here today on this 13th Global Conncet program. Thank you very much, Dr. Mithila.
Thank you. Thank you for the nice words. Everybody was so friendly, so nice. I'm so glad I did this. Yeah. Thank you.
Thank you. Thank you.
Thank you, madam. Thank you, sir.
Sure.
Thank you.
I shared my email in my presentation, Dr. Rao. If you any student interested to contact, please share my email with them. I'm more than happy to answer their question.
Sure. Thank you.
Thank you.
Thank you very much. Now the program is ended.
All right. Bye, everyone. Take care.
Bye, madam.
Bye.